So hi again, my name is Ijeoma Obi. I'm from the Bangor High School STEM Academy, and my project is the synthesis of an azobenzene-based cholesterol mimic for gated ion channel modification. So the research question here was, how can an azobenzene-based molecule be synthesized to mimic the structure of cholesterol? And the reason why this is of great interest is because of pharmacy and photopharmacology. So photopharmacology is the process as to which light is able to change the conformational shape of living systems and therefore change their functionality. And these molecules are what we refer to as photo switches in terms of organic chemistry. So as you probably already know, cholesterol has this bulky storage structure, and cholesterol has this amazing property where it's able to change the permeability of ion channels without directly interacting with the ion channel itself. So cholesterol is able to interact, as you can see from the diagram over there, similar to a lysolipid, where it's able to open and restrict ion channels by simply entering and exiting the cellular membrane. So the research target here was to try to make something similar to cholesterol in order to be used as an ion modulator. Ion modulators are classified as drugs that are able to change essentially the permeability or the amount of a substance that goes in and outside of an ion channel. So for example, some diabetes medications are ion modulators that can control the amount of calcium that is going in and out of an ion channel. And so that was the research target, was to make a simple, easy ion modulator that can be controlled by light, making it a simple active substance to be used in different shapes and forms. And in order to classify this molecule, I use UV-vis spectroscopy and proton NMR spectroscopy. So if you look down to the materials section, you can see that the chemicals I used did include HCl, MEOH, isoamyl nitrate, and etc. The solvents that I tested to make sure that the molecule, the azobenzene, was similar to cholesterol included methanol, acetone, ethanol, and DI water. And the equipment and programs I used are also listed below. So if we move on to methods, the reactions to make this ion-based cholesterol mimic were broken down into two parts. The first part being an azo-coupling reaction. And this azo-coupling reaction is what allows for the main azobenzene structure to be made. And this is where two diazonium salts are coupled together by an N-N bond or an N double bond. And then the second part was the substitution reaction in order to add in an aliphatic tail so that the azobenzene was even closer to that of the original cholesterol structure. So quickly going through the methods here, UV-vis spectroscopy was used simply to characterize the molecule. Solubility tests were also used to see if it was similar to that of cholesterol. Photocycling was done to see if the molecule was able to change its conformational shape. And so conformational shape overall is to see if whether the molecule can be active or inactive by changing its shape and form. So sort of like a power switch. And then proton NMR spectroscopy was used simply to ensure that the actual target molecule was synthesized. So looking at the results here, part one of synthesis, we can see that the final product, the substitution reaction, there was a 45.1% yield, which indicates that this molecule was on the most part synthesized. Solubility tests show that the molecule was soluble in organic solvents except for water, which is very similar to that of cholesterol, as it's also quite soluble in organic solvents except for water because it's somewhat of a lipid structure, making it an ideal molecule for a mimic. The UV-vis spectra observed from methanol at 480 nanometers shows the wavelength and absorbance. And this helped me move to the photocycles. And the photocycles go back and forth in terms of absorbance, which indicates that the shape of the molecule was changed because the absorbance changed depending on the wavelength of light, UV light or visible light that was shone onto the molecule. Proton NMR spectroscopy was done, and both amine groups indicate that the molecule was fully synthesized. The photocycles observed in DMSO also fully prove that the molecule can change its shape and is able to change its structure when irradiated on light. And additional modifications, just other variations of this azocholesterol were made. So what can be concluded from this research is that photo switching the azobenzene is possible simply with UV light and visible blue light. The proton NMR fully shows that this azocholesterol mimic is possible and has been produced. And the azocholesterol is a mimic towards cholesterol with similar functional groups. So future work, I continue to plan on making different molecules similar to that of cholesterol. I continue to keep on testing more ligands. And overall, in terms of world applications, this molecule really can be used as an ion modulator to possibly help with drug delivery, especially for drugs such as diabetic medications. My references are seen below, and at this time, I'd be happy to entertain any questions.
Wow, that was fascinating. So, has this azobenzene compound been used in human pharmacology with drugs that are in existence right now? Is there safety data about it?
There is some safety data about it. However, azobenzenes have mostly been used, especially when it comes to food science, because azobenzene, it's red in nature, this orange reddish substance. So it's been used especially when it comes to food dyes. And it's also been used in clothes. The molecule was found about 200 years ago, but recently, in terms of the literature that I've researched while doing this project, it's been about 50-ish years since people have started realizing that azobenzenes could be used simply more than just a food dye or a clothes dye.
So how does the azobenzene compare to cholesterol in terms of size? Part of the rationale of cholesterol in terms of the membrane dynamics is that it sits and it's very planar and very stiff inside of one side of the lipid bilayer, and therefore it causes changes in the dynamics of the plasma membrane. Is this a similar size, and therefore you would predict that it would actually be able to embed itself in the membrane in a similar fashion?
So to answer your question, azocholesterol has two forms, right? So the trans state, which is very similar in shape and size to that of the cholesterol, and then the cis state, which is when it's changed when irradiated with UV light, and that's when it changes its geometric formation and seems to be bent. So to answer that question, the azocholesterol in its normal trans state would be able to interact with the cell membrane. However, more research would be needed, well, when the lab is available to really look at how a cis azocholesterol would interact. However, when it comes to the physical groups, the one thing that makes an azocholesterol so interesting is that it doesn't need the bulky steroid structure to necessarily interact with the cellular membrane. So when working with variating this molecule to be similar to that of cholesterol, adding in the aliphatic tail, but also making sure that the photo switch is within the molecule allows for some variation, but also allows it to be similar in terms of the functionality of cholesterol in the cellular membrane.
So just for clarification, in terms of the utilization that you're proposing here, would you have the azocholesterol be embedded in the cell membrane, and then you would target it with light to then cause the conformational shift, or would that be something that would be done prior to delivery and its association with drug delivery?
So to answer that question, it can be done in two ways. One way that I've been thinking about is the first way that you've just said, which is the whole idea of having the azocholesterol already in the cell membrane, and then after irradiating it with light. However, naturally in the human body, I'd assume it'd be pretty difficult to just get cells out in the open and shine like a flashlight on it. So I think the whole idea overall is to have the azocholesterol be given to the patient, and then after which, when ingested, So cholesterol, although it may seem like it can only be transformed by light, like many conformational changing molecules, just some form of energy would be able to allow it to change its structure. So possibly as well, temperature can also allow for this molecule to change. And in that sense, that can be a way for allowing it to expand and dilute overall just the structure of the cellular membrane, so that that way the ion channel is able to open and close and change its permeability.
Great. Thank you very much.
Thank you.
Hi, this is Ann. Very nice presentation. What would you say are the biggest advantages of this over the current modifiers that are out there for ion channels and drug delivery?
Well, I think one advantage is when it comes to azocholesterol is just the azobenzene itself. Azobenzenes it's a material that's used overall globally, and it's used in almost everything that we use today. So as I've already said before, it's used in clothes, it's used very commonly in our food, in candy, and even in drugs. For example, some drugs that are coated with red outside of them have some form of an azobenzene pigment. So overall, azobenzenes, I think in terms of cost analysis, would be cheaper for using as an ion modulator than other ion modulators currently, because the molecule and the material itself is overall used more often. So the fact that it's already out there, I think, will give people the opportunity to use it at a cheaper price than what's already being used for ion modulators.
And do you have any reason to think that this would allow for a faster drug delivery over other modifiers?
So the reason as to why I believe it would allow for faster drug delivery is what I saw while working in the lab on this project. So under UV light or some energy similar to UV light, the molecule was able to change its shape in 5 minutes. And then reverting back to its original shape, it only took 5 minutes as well. So I believe in terms of energy transfer, in order to allow it to change its shape back and forth in terms of conformational structure, I think it is possible to have faster drug delivery when it comes to this azocholesterol.
And do you have where would you start in terms of testing this in cells? Like what cell model would you use?
In terms of testing it in cells, I would try to simply have a cell, nothing special about the cell at all, but overall the main thing that has to be tested when it comes to this cell is just the cellular membrane and possibly just one ion channel. So overall, in terms of making cells to allow this molecule to be tested, the cell overall doesn't have to be anything unique or special. It could be any type of cell as long as it has an ion channel and a cellular membrane that allows for the azocholesterol to interact with those lipids so that we can see overall the changes in the ion channel.
Great, thanks so much.